All right, let's continue. This is part 2, Campbell's 7th edition, chapter 48. So here's the basic structure of a neuron. It's a generic neuron. There's different shapes and different types that we'll get to shortly, or we'll talk about later. But here's your generic structure. So every neuron has a system of dendrites, which collect information from a variety of sources. A cell body, sometimes called the soma. And the cell body is the structure that is much like every other cell: mitochondria, nucleus, Golgi, and so on. And then you have one axon, which is the process that emanates out from the cell body and carries the information away, carries the action potential away. So when we talk about action potentials and how they work later, we're largely focusing on the axon. We could also be talking about the membrane of dendrites, but we'll be largely focusing on the axon. You'll also notice that this axon is showing a myelin sheath. Not all axons have a myelin sheath. Some are unmyelinated, but in general, these marshmallow-like looking things are myelin, and there's gaps in between, and those gaps are called nodes of Ranvier. This is the presynaptic cell. It synapses with the postsynaptic cell, where the axon terminal, or the end of the axon, synapses with the dendrites of the postsynaptic cell. Now, this could be a neuron, this could be a gland, or this could be a muscle. Now, a lot of the attention in the nervous system goes to neurons, but in fact, they are not the most numerous cells in the nervous system. They may be classified as the most important, if you like, but the variety of other cells that support the nervous system and play very important roles, they're called glial cells, or neuroglia, and they maintain structural integrity. They provide help support nutrients and strength and so on. So we'll go through a few of those here now, just some general ones. Here we have astrocytes. Astrocytes they're shown in green, and they provide structural support for neurons, and they help to regulate extracellular concentrations of ions and neurotransmitters. Astrocytes, their name comes from the fact that they look like stars. Oligodendrocytes play an important role in the central nervous system. Oligodendrocytes make up the myelin sheath that wraps around the axons. They're actual cells that get pulled and wrapped around, kind of like a blanket, if you like. And the other type of cell that creates myelin sheath are called Schwann cells. Schwann cells make up the myelin in the peripheral nervous system. Two different cell types, depending on location, that make up or are the myelin sheath. Here you can see an actual picture under the microscope. All right, 48.2. So maintaining a membrane potential, which is essentially a difference or voltage across the membrane. This is done by ion pumps and ion channels. At rest, the cell is negative inside compared to the outside. Now, there's a lot of attention, or students often ask, if we're talking about membrane potential and focusing on the ions sodium and potassium, how is it that the inside of the cell is negative? Well, there's two ways to describe this, both partially correct, although I like the the latter is more accurate. So the former, or the first way to describe it, is simply that the inside of the cell is negative relative to the outside. So if you compare the charges, the outside is more positive than the inside. In fact, the better answer, the one that I prefer, is that the inside of the cell incorporates all the nuclear components as well in the cell body. And because of that, there is DNA that plays a role, and DNA is very negatively charged. It's very polar. In fact, every nucleotide of DNA has a -2 charge. And because of that, that makes the inside of the cell very, very negative. So at rest, the membrane potential of a neuron is not transmitting any signals. There's a gradient set up between ions, and everything is just stationary, or let's call it, there's a dynamic equilibrium with some of the ions, but everything is just kind of status quo, nothing happening, but it's prepared or ready to fire. So what we can do is there's a mechanism. We use a microscope, and you can do this in labs in college. I remember I did it in college in a neurobio lab. And there's a microscope that uses an XYZ axis to move a teeny tiny electrode. It's a glass electrode, and in the middle of that glass electrode is a teeny tiny metal strip. And you just insert that into the membrane of the cell, and it's connected to a computer that's going to measure the voltage. And at rest, this measurement is showing up as -70 millivolts. Okay? And then you can do all kinds of manipulation things to it. to mess with the voltage and see what happens, and this is partially how we understand how an action potential works. Now, the at rest here, we're talking about, once again, the outside of the cell versus the inside of the cell. The outside of the cell, here you'll notice a voltage difference of negative 92 millivolts. In this inner chamber, there's 150 millimolar of potassium chloride, very high potassium. Here, very low potassium. Okay? And here they're showing you sodium chloride. The outer chamber has a very high concentration of sodium chloride compared to the inside. And in fact, the difference between the two, much like a cell, is about 10 times the gradient that's generated. The sodium gradient and the potassium gradient in opposite directions have a difference of 10 times. Now what you're going to notice here is there's a couple different types of potassium channels that we'll get to. There are potassium leak channels, and there are potassium voltage-gated channels that play a role in repolarization. Potassium leak channels, there's a lot of them, and they're open all the time for potassium to leak from the inside of the cell out. Sodium, on the other hand, there's a few sodium leak channels, but the majority of them are sodium voltage-gated channels. And you'll also notice here, once again, you can see the difference. This is positive 62 millivolts, this difference in charge, compared to this one being negative 92 millivolts. We won't get too much into that, but you do notice that. So a neuron that's not transmitting has many open potassium channels. These are the potassium leak channels that I described. And there's fewer open sodium channels. These are sodium leak channels. They're just pathways for sodium to pass through, but there's very few of them. Okay? The diffusion of potassium and sodium through these channels creates a separation of charges. This is the actual membrane potential that is occurring at rest, at negative 70 millivolts. And we'll take a pause on this one.